Hello everyone in Montreal. I'm Charlene from the University of Hong Kong area in ecology and biodiversity. Due to the issue with my visa application, it is a huge pity that I couldn't attend the meeting and give this talk in person with you. However, I really appreciate the chance to present our current progress on the mutation rate project to everybody at this symposium. I welcome all the questions and discussions if you are interested in this topic. I would also like to thank Dr. Bathomi and Dr. Krosbach. I hope I didn't pronounce it incorrectly for your amazing organization with this symposium and give me the opportunity to present our work on the de novo mutation rates in sticklebacks. And let's start with the term de novo mutations, which I believe everyone here is pretty familiar with. They are mutations that newly arise. So when one has data from a parent offspring trio, these mutations should only be observed in the offspring, but not in their parents. They are rare but transmittable over time, serving as the ultimate source of genetic diversity and the potential inherited diseases. And therefore, the rate of them is also essential as a key scaling parameter in genetics for the demographic or the phylogenetic inferences. Given the importance of the mutation rates in evolutionary genetics, it has historically been a crucial task for geneticists to estimate mutation rates through various methods, and this includes mutation accumulation methods, which estimate the rates of newly occurring mutations by allowing them to accumulate for generations until they can be easily observed and quantified. With recent advancements in sequencing technology and computational tools, a more straightforward method is to estimate de novo mutation rates directly based on the pedigrees, based on the parent offspring trios data. This involves comparing the genomes of the parent and the offspring to identify changes like SNPs, indels, or structural variants directly. And this method doesn't require any assumptions about generation time or fossil calibrations. And it can provide more information about the genomic locations and the spectrum of the mutation types, those features of mutations as well. However, high sequencing depth is needed to ensure the accuracy of these estimates, especially if using the short-read dataset. This slide shows a figure from our paper providing the first estimate of mutation rates in non-spine sticklebacks. It summarizes various mutation rates estimated using this method in 30 species covering 30 studies covering 86 species, including the new data points from a quite significant paper published last year in Nature. It shows that the mutation rates are now available for numerous species across the tree of life. This also includes quite a lot of non-model species, as you can see. Although, of course, this figure doesn't include all the studies published so far. So here I provided some more publications with using this specific method estimating de novo mutation rates, including some estimates from plants, some additional estimates from the arthropods, and new studies published after us from some other mammals, including the marine species whales, and like the other marine species, the algae and starfish. Many discussions in these studies have helped us better understand the mechanisms behind these numbers. For example, how hibernation from this bear paper and the haploid diploid cycle from this algae paper, and in fact the population sizes or the poverty age can influence the mutation rates, and also some discussions about the technical issues related to the reference biases influencing the accuracies of the estimates from this paper published this year. Also, some discussions are happening around and some insights like if mutation rates can explain the low cancer rates in the gigantic mammals as well. Those are pretty interesting discussions happening nowadays. However, the estimates of germline de novo mutation rates were still not available for the supermodel species in ecology and evolutionary biology in those sticklebacks before our first paper coming online. These small spiny tailed-fish are widely distributed in the northern hemisphere, and the three-spine sticklebacks are pretty well known for their repeated adaptations from the marine to the freshwater systems. And the standing genetic variance is repeated selected after isolation in the freshwater environments, causing the differences in defensive and feeding morphologies between the ecotypes, such as the repeated reduction of the pelvic spines. Nine-spine sticklebacks at the same time have also emerged as a model system over the past decade that distributed around the North Pole in both marine and freshwater systems as shown in this red area. And their sister species in Punkitis genus are quite diversified, sometimes having different sex chromosome systems even. And the genetic diversity is highly differentiated between individuals living in the marine versus freshwater systems, as shown in these blue bars. Nine-spine sticklebacks has relatively small genome size at about 450 megabytes. The reference genome of them is well defined as well with the annotations available. And the latest version of the Western European lineage, published in December last year, was provided by the Tree of Life project. And a version of the Eastern European lineage was published by our lab earlier this year. These two lineages of the European nine-spine sticklebacks also have different sex determination regions. Up to date, actually, there aren't many thorough comparisons of the de novo mutation rates at the intraspecific level, where the genetic background can be mostly controlled for any comparisons. Also, the interspecific studies in fish species specifically, comparing the rates and the mutation patterns. And therefore, we aim to address these research gaps with our large pedigree data set. And I'm going to focus mostly on the second research gap in this talk, which we are currently working on. We sampled 9 spine sticklebacks from 4 pound and 4 marine populations of the Eastern European lineage. They were bred into 4 to 5 pedigrees of 2 to 3 generations. And after confirming the parentage relationship of these individuals, 447 samples consisting of 367 trios were included finally in this data set. And our workflow for identifying these mutations is shown in these slides. We followed basically the guidance provided by the Mutation Named On paper, but with some extra analysis like the IVD tests that I just mentioned to confirm the parentage relationship before doing any further analysis. And using the read counts from the final band files or multiple replicates of the virtualization check in IGV to confirm the false positive rates. Because of the time, I won't go into too many details of these pipeline, but any questions or discussions are surely welcomed through emails later if there's any. We provided our first estimates using preliminary data sets from 2 marine populations shown in this cycle. This involved 10 inbred and outbred pedigrees, including 128 samples And we finally identified more than 300 unique de novo mutations in total. And the final germline de novo point mutation rate was 4.56 times 10 to the minus 9 per base pair per generation. This estimate resides in the middle of all fish estimates up to date. Most mutation rates in fish were estimated to be lower than 10 to the minus 8, except for one in guppies published last year. And the lowest estimates came from the Nathaniel Sharp. We potentially provided the most accurate estimates with the largest data set and the narrowest 95% confidence interval. And after that paper, we managed to obtain data from the remaining 6 populations and estimate their de novo mutation rates. And the average was respectively 5.68 and 4.21 times 10 to the minus 9 in freshwater and marine populations. However, we didn't observe an obvious difference in de novo mutation rate between 2 ecotypes. And this slight significance shown here is more likely driven by the hypermutable outliers from one single family. We estimated the alpha value as well, which is the paternal to maternal ratio in terms of which parent the mutation is inherited from. Our estimate was about 1, whereas in the other vertebrates, this value can be quite high, indicating a different level of male bias in mutation inheritance. However, in sticklebacks, we even observe a slight female bias in the freshwater ponds. And another interesting fact is that we estimated the percentage of shared mutations among 4 siblings at about 15% on average. This is the largest among all the reported studies highlighting the specialness of fish species in mutation inheritance compared to other species. Coming to the drift-bearer hypothesis, which suggests that selection is more restricted by genetic drift in small populations, and therefore we expected to see a higher mutation rate in those smaller populations. Factors like the life history traits, the inbreeding level, the reproductive longevity, the generation time should also be correlated with this pattern. We estimate the effective population sizes of the 8 populations using MSMC2. The NE is obviously higher in marine populations as indicated by these blue trajectories, and the marine populations have generally 10,000-fold higher NE than those freshwater ponds in yellow, but the mutation rates are not significantly lower as what has been hypothesized. And what we can notice is also that although the pound have similarly small population sizes, their mutation rates are more variable than the marine populations. To increase the power of our analysis, we added data from sister species in Punkitis genus and the star species that I just mentioned, the 3-spine sticklebacks. Including these data from related species allow us to explore the relatively broader patterns, providing a comparative framework of this topic. But we still failed to prove the drift-bearer hypothesis, whether comparing mutation rates with the endpoints or the points along these trajectories until about 10,000 years ago, which is when freshwater ponds were reported to have started isolating from the marine system. We are also trying to utilize the other NE estimators at the moment, like the GON or current NE, to provide wider comparisons. But at the same time, it led us to think about some other possibilities of determinants of mutation rates. This include the parental age of those well-caught fish, of those parents, All the otoliths of these wild-caught fish were collected, and our collaborator, Dr. Himbrand, helped estimate the fish age by dyeing those little tiny otoliths and counting the rings on those. And the parental age can influence mutation rates, with older parents contributing to more mutations to their offspring. However, we only saw a weak correlation between maternal age and the mutation rates, and this is even not true for the correlation with the paternal age. The freshwater individuals tend to breed later than those living in the sea in 9-spine sticklebacks. This is the opposite to 3-spine sticklebacks. But there is an overlap between the breeding ages at 2 years old, when the maternal age is controlled for. Mutation rates appear higher in freshwater pedigrees than those living in the sea. So as you may see, these processes quite complex, with various factors influencing dynamic mutation rates nested within each other. Even with our big data set, the power is still not enough when controlling some factors. But it provides a nice foundation, of course. And we are continuously refining our analysis by incorporating additional data sources and developing more sophisticated models. Hope these further explorations will enhance the robustness of our findings. Also, your suggestions are highly valued and welcome if there's any. Following that, we are also trying to explore the pattern of localized mutation rates throughout the genome, and we did detect a significant correlation between mutation rates and recombination rates throughout the genome, in marine populations specifically. This is not surprising because the linkage map applied in this estimation was established with a marine population from the Eastern European lineage published last year. So the pond recombination pattern could be pretty different and diversified among different populations. And the recombination hotspots may be particularly prone to the mutations, and they are reported to be located within the promoter-like regions throughout the genome in species lacking the functional PRDM9 gene, which include the sticklebacks. We would like to test this specific prediction as well to see if mutations are more likely to be located within these areas, including the CPG islands, for example. In addition to these directions that I just mentioned about, interspecific and intraspecific comparisons of the mutation spectra also interest us, which will be our next goals following our current findings. Here comes to our take-home messages from this talk. We provided the first dynamo point mutation rates in sticklebacks with estimates from different ecotypes and species lineages. We observed different patterns of mutation inheritance in fish compared to the other organisms, with less male biases and more shared mutations. However, we haven't proved the drift barrier hypothesis with our current data set, but our findings highlight the complexities of the mutation rates, evolution, and the need for further research to understand the underlying mechanisms. And you are welcome to scan the QR code to learn about our first estimates of mutation rates in sticklebacks and other features of these mutations that I didn't cover in this talk. Here, I would like to thank my supervisor, Professor Johan Barilla, my collaborator, Professors Skirup and Dr. Himbrand, for their advice and support throughout this journey towards here. And Also, I would like to thank Dr. Brightmold, who collected and developed the pedigrees. I also appreciate my lab mates and friends, especially Dr. Reid, Dr. Sands, and Dr. Liu, who provided important insights into this project. And thank you all so much for your attention to my talk. I really wish I could have given this talk in person rather than virtually, but I still hope you enjoy the conference and this presentation. Please feel free to email me if you are interested in any discussions or collaborations related to this project. You may probably also leave comments under the video after it being posted on YouTube. And cheers, everybody, and wish you enjoy the rest of your day. Thank you. Goodbye.
All right. Hi, everybody. I'm going to stand here awkwardly because I can't actually see my slides on here, but we'll all look together. So I'll talk to you about some maybe alternative evidence in support of the drift barrier hypothesis. Love the discussion around this. We're thinking about mutation rates in haploid and diploid cell types of different yeast species. And so, of course, we're here because we're interested in mutation, and mutation rates vary. I think we're all pretty convinced that they're quite variable, and the question that we have is why? Evolutionarily speaking, why do we see this variation? Let me suggest that we can kind of put hypotheses in two buckets here. One possibility is that maybe natural selection is working all the time to optimize mutation rates, but that it's doing so in different ways in different organisms. Maybe different organisms have different needs or propensity for beneficial mutations, and that's helping to drive mutation rate evolution. Maybe there are different trade-offs between genome maintenance and other traits, and those kind of life history trade-offs are operating in different ways in different organisms. And so in a kind of a quantitative genetic sense, we could say that that would predict that mutation rate is under stabilizing selection in any given species, right? That there's some non-zero optimum mutation rate, and selection is kind of trying to keep the population there. An alternative idea is that perhaps in general, we know there's always exceptional cases, but in general, perhaps natural selection always favors lower mutation rates. Maybe natural selection is trying to push mutation rates to be as low as possible, but there are limits to selection. We know this from various kinds of evidence that natural selection is not all powerful, particularly in small populations, but this is true for any finite population. If a given allele has a small enough effect on fitness in a particular finite population, that allele will be effectively drifting around rather than becoming fixed or lost due to natural selection. And so if that's, another way to frame this was that weak mutator alleles that have a weak effect on fitness could become fixed by genetic drift. So even though natural selection is favoring 0 as a mutation rate, we see higher mutation rates. And this is the drift barrier hypothesis, as we heard about in the previous talk. And I guess I would want to note also that I'm not sure that weak mutator alleles have to become fixed by genetic drift necessarily. They just have to be drifting around, and they could even be at intermediate frequencies in populations. And we don't, I think, know the answer to that yet. There's some evidence for the drift barrier hypothesis, as has been discussed. So across a broad phylogenetic scale, we do see this negative relationship between effective population size and mutation rate, where the mutation rates are lower in larger populations, and this is seen as a basic form of support for this idea. But what I want to do is maybe take a different tack here. I think that the drift barrier hypothesis in that body of theory makes more predictions about mutation than just what we expect to happen in different taxa with different effective population sizes. The drift barrier hypothesis really says that the effectiveness of selection can be limited in some circumstances and less limited in other circumstances. And so if the drift barrier hypothesis is correct, we should see differences in the effectiveness of DNA repair pathways that are used very frequently or very rarely, we should see differences in common versus rare cell types in mutation rates. And so what we did to kind of tackle this idea was considered haploid and diploid yeast cells. I'm going to refer to some previous work I've done for comparison and then some new data from a second species, but our previous work involved the budding yeast Saccharomyces cerevisiae, and it's important for me to let you know what I think about their general lifestyle for this talk. So if you know about yeast, you're probably aware that we can grow them in the haploid form or the diploid form, but they're really a diplontic organism. This is a thing that prefers to be diploid in the wild and has almost certainly been diploid for the vast majority of all its cell divisions through its evolutionary history. There's a couple of reasons that I think this is the case. So natural strains don't do meiosis very often. They're mostly growing asexually. And when they do meiosis, they form a tetrad of haploid cells, and those haploid cells are kind of bound together in most cases. And when they germinate, when conditions improve and they can germinate, they'll most likely mate with one another. We could think of this like selfing. So how many generations have they been growing asexually in the haploid state? 0. They didn't grow asexually in the haploid state, they just mated. What happens if we were to get a lonely, maybe I don't need the microphone. What happens if we were to get a lonely haploid all off on its own? Oh, sad. The mating type switching process allows it in one cell division to produce 2 daughter cells of opposite mating types, which then will mate together immediately. This is kind of like a reproductive assurance tactic, perhaps. And so we don't expect this haploid state to last very long either. They have a high propensity for mating. And so you might ask, well, what's happening in lab strains where we have haploids and diploids? Well, the vast majority of lab strains are genetically manipulated so that they cannot switch mating type. And what that means is that if you take a single A or alpha cell, you can grow a haploid version of that strain without them mating with anybody, and they can maintain haploidy that way. And so I think we should think of Saccharomyces cerevisiae as a diploid organism for the purposes of this question. And so what we did was made a prediction that in this diplontic species, natural selection has had very little opportunity to act on, let's say, any weak mutator alleles, or really any mutator alleles that might be specific to the haploid state. If there was an allele that affected haploid mutation rate, natural selection would not see it in this organism. So we predict a higher mutation rate in haploids. That is what we found. There's about a 40% higher single nucleotide mutation rate in haploids than in diploids. We also found that was the case in the mitochondrial genome. No difference discovered here for indels, but those are a rarer type of mutation. Diploids do have a higher propensity for big structural changes, so there's a potential role of genome architecture there, but for the most part, I think this prediction held up. But there's kind of a problem, as I see it, with this argument, which is, what if haploidy itself is mutagenic? What if it's harder to repair your DNA when you only have one, or you typically just have one copy of each chromosome available? Perhaps haploids are just inherently more mutable than diploids, and this has nothing to do with their evolutionary history. And so what we wanted to do was kind of test that idea by looking at an alternative species. And so I'll start comparing our 2 species here, so that we've got our budding yeast, which are diplontic and spend most of their time as diploids. But there are other yeast species, such as Schizosaccharomyces pombe, the fission yeast, which have the opposite kind of lifestyle. They are haploid almost all the time. When conditions get bad, they go ahead and mate with one another to form diploids. The diploids sporulate immediately, and then upon germination, you've got haploids again. And so what I'm kind of trying to show here is that these guys have opposite lifestyles, mostly diploid versus mostly haploid. And so what we did was a mutation accumulation experiment in fission yeast. MA experiments in fission yeast have been done before, but just in the haploid form, and we wanted to do it in both the haploid and the diploid form. How do we prevent, so in budding yeast, we have to stop them from mating to maintain the haploid state, which we do by knocking out their mating type switching. We have a different challenge in fission yeast. We have to stop them from sporulating when they're in the diploid state. It turns out there's an allele that does this. It's a single nucleotide change, in a transcription factor, and these we can make cells that are normal diploids in every way except that they can't sporulate. And so we just take haploids, let them mate with themselves. We have a genetically identical haploid and diploid strains, and the diploids will stay that way, or at least they should. We've got 50 lines of each type. Based on cell counts throughout mutation accumulation, the haploids are growing just a bit faster than the diploids, perhaps not too surprising, but really they're quite similar in terms of their fitness, and we'll use those values when estimating their mutation rates. Perhaps I should briefly say that mutation accumulation is a style of experimental evolution where we're repeatedly bottlenecking strains so that natural selection during our experiment will not play a major role in which mutations become fixed or lost, allowing us to kind of see mutations accumulating at their natural rate. I should first say that when we do an experiment like this, setting up haploids and diploids, we want to make sure that at the end of our experiment we still have haploids and diploids and that they didn't change ploidy type, which is known to happen in budding yeast as well. These guys seem to do it even more often than that, so we have 6 strains that went from diploid to haploid through some unknown mechanism, and 1 that went in the opposite direction. This is kind of neat, and I would like to hear any suggestions for how to investigate this further, but for the purposes of this talk, we're just going to exclude those lines, because their ploidy history during the experiment is mixed, and we wouldn't have a clear prediction for what their mutation rates should look like. Okay, so now I'm going to compare the budding yeast data to the fission yeast data on the same plots, just so we can kind of put them side by side. And our prediction here is confirmed. So in both species, the mutation rate is significantly higher in the rare cell type. So this is evidence that maybe the drift barrier idea is playing a role, and not so much that haploidy per se is mutagenic. Right? If haploidy was mutagenic, we would see both red bars being higher. We actually have a pretty similar magnitude of the difference in this case. The fission yeast have an overall lower mutation rate, and that's not too surprising given that, as far as we know, they have a higher effective population size overall. We, of course, looked at the mutation spectrum as well. So the single nucleotide spectrum differs between haploid and diploid budding yeast, and it also differs between haploid and diploid fission yeast. We also find some homozygous mutations in the diploids, which indicates that loss of heterozygosity events are occurring in these asexual diploids, which are also known to happen in the budding yeast. As far as I know, this is the first evidence for that, and we're investigating those further. They seem to be sort of further from the centromere than you would expect by chance, and exactly what you would imagine double-strand break repair processes to give us. A couple more results here. So insertion-deletion mutations, we didn't see a difference for the budding yeast, but we do see a difference for the fission yeast in the same direction as for single nucleotide changes. Their rates of indels are much higher. We knew that already. And they also have an insertion bias, but the insertion bias seems to be depressed in the diploid versions of the fission yeast, which is interesting. We should be concerned about natural selection affecting our results during mutation accumulation, particularly in this kind of comparative framework where we're looking at haploids in the one case and diploids in the other case. What if we see fewer mutations in haploids because recessive deleterious alleles are being kicked out by selection in the haploid case but are persisting in the diploid case? And so we look for this by comparing the frequency of mutations in protein coding regions or in synonymous, non-synonymous ratios, and basically we don't see any evidence that natural selection is driving our results here. Okay, so to summarize, in these two species, the rare ploidy type has a higher mutation rate. That's consistent with the idea that natural selection has generally acted to reduce mutation rates, but that it can be prevented from acting effectively on mutators that are specific to a rare cell type, because simply natural selection can't see those cases very effectively. And so my take-home message is that maybe this mutation drift balance has an important role in mutation rate variation within species as well as across species. So I think there are more opportunities to kind of test the drift barrier idea in other ways like this. I should acknowledge my co-authors. So Kevin Bao is a grad student in my lab. Rutuja Gupta is an undergrad student in my lab, and they have both worked really hard on this project, and happy to take questions.
Okay, thank you very much for the opportunity to talk. I'm excited to tell you about some of the work we've been doing over the last few years and recently in the green alga Chlamydomonas, and specifically looking at these sort of large structural mutations that we know are very important in genetic disease and evolution, but have historically been very difficult to study. So my outline today, I'll just very briefly introduce why Chlamydomonas green algae are good models to study evolution and mutation in particular. And then I'll sort of go through two results. The first part is published work, but just to set up the final part, which is looking at using long-read sequencing to call sort of the full spectrum of structural mutations, and then using some short-read data, going to more genotypes to specifically look at transposition rates of mobile elements. So Chlamydomonas reinhardtii is a model system. It's used a lot outside of evolutionary biology, specifically in photosynthesis research and cilia biology, but it's quite good in the lab for experimental evolution as well. So it's a tractable system. It's generally haploid. It does have a diploid form, but that's just a transient zygote. And we can control sex, so we can grow it clonally, we can induce sex. Has all of those nice benefits similar to the yeast systems that we just heard about. And one thing that is really fantastic about it is it has a fairly representative genome structure, so sort of similar in size and complexity to Arabidopsis or Caenorhabditis, over 100 megabase genome. And my favorite thing about it is it has an incredibly diverse repertoire of transposable elements. So the data I'm going to talk about today actually all goes back to a sort of series of experiments that were performed more than 10 years ago now at the University of Edinburgh. And this is, of course, this sort of classic mutation accumulation experiment where we grow Chlamydomonas on agar plates. A random colony is picked, transferred, diluted, new colonies grow, another colony is picked, and this is continued for many serial transfers. And for these experiments, generally speaking, they were continued for over or just up to 1,000 generations of growth. And I say this key point is that these are continuously bottlenecked, going back to a single colony, every single transfer. So you're minimizing selection and getting a fairly representative view of mutations. And the largest experiment of this type that was done was on 6 wild genotypes of Chlamydomonas. These are all from different parts of North America, and Chlamydomonas actually has really incredible genetic diversity. These all differ by more than 2% at the SNP level. And there's, as you can see here, this is the single nucleotide mutation rate for a number of replicate lines, extensive variation among genotypes and also within genotypes in the single nucleotide mutation rate. Talking of strange strains, the one on the end has been in the lab for a lot longer than the others, so it's possible that that's some sort of strange laboratory thing that's occurred. A second experiment that was published quite a lot of years later was done on a different species. This is the closest known relative of Chlamydomonas reinhardtii, but it's probably 10 to 20 million years away in evolution. And it's kind of cut off, actually, but different scale here because it's a really extreme hypermutator. And you can see some of the hypermutators in the other lines as well. So again, these are probably things that have occurred during the experiment where there was a mutation that impacted DNA repair, and you got this accumulation of mutations. Okay, so the first thing I'm going to talk about is all published work, and this is where, at the end of my PhD, together with a postdoc in the lab, we asked if we could go back and get all these strains out of liquid nitrogen and sequence some of them with long reads. So we didn't have money to do everything, but we chose 2 genotypes, one with a lower and one with a higher single nucleotide mutation rate. We sequenced the ancestor, so we made a reference genome specifically for this purpose, and then we sequenced either 4 or 8 of the mutation accumulation lines. And this was done at sufficient coverage. These are haploids, and this highly accurate long-read sequencing, you don't actually need very much to essentially make near-complete assemblies. And I'm going to completely skip over the methods, but we basically used a number of tools, both by aligning the assemblies to the reference and also aligning the reads to call structural mutations. And between 2 of us independently comparing, we curated everything, so we have a kind of high confidence data set of structural mutations. And this is a summary of this data. So I'm scaling this by sort of structural mutation events per genome per 100 generations, just so it's a little bit easier to interpret. And you can see again that we have extensive variation among these two genotypes. And in particular, transposable element insertions were the largest category of structural mutation. So sort of 0.6 per 100 generations up to 1.5 per 100 generations. But there are also a significant amount of deletions and duplications, again with differences between the strains, and many, many more rearrangements, so translocations, inversions in one of the genotypes versus the other. And if we compare this to smaller mutations, single nucleotide mutations and indels, in one strain it was around 6% of mutations overall were structural, while in the other it reached 12%. So a large proportion of mutations were structural. If we dive into those transposons, there's a lot more information than you need here, but this is basically just a number of different types of transposable elements over the different replicate lines that we sequenced with PacBio. And you can see that there's a really great diversity of transposable elements active in one strain. This is the one that had a higher rate of transposon insertions. And again, you've got a lot of variation between the lines. While in our other genotype, we actually only had one single element active. It was active at quite an appreciable rate, but compared to the strain that had a higher rate, we had this kind of quite wide spectra of different transposons. In our other strain, we just had one single retrotransposon that was active. And the final thing I want to say about this data is that a lot of the other mutations that we saw were actually mediated by transposons. So there were sort of 3 different transposons that were involved in this, but just one example, this is a reciprocal translocation. You can see the ancestral chromosomes in purple and green, 9 and 11, and then in our mutation accumulation line, you have this translocation. And when we go in and look at the breakpoints, what we see are insertions of this particular type of element called a krypton. It's a really obscure, as the name suggests, transposon. This is the first time anyone's ever seen one of these jumping. And they're actually recombinases, so they basically perform recombination between a terminal sequence and a target sequence in the genome. But what we saw is that when we went in and actually looked at those breakpoints, those translocation breakpoints, we saw a target sequence, we saw an insertion, and then we saw the translocation. So we think that this recombinase is actually able to mediate recombination between chromosomes. So I only just put this here as an example just to show that many of our transposon insertions were not simple. It's not just a transposon going in, it's a transposon going in and then mediating some other large change as well. So just to summarize that part, the published work, the rates of transposition and other structural mutations, I think we can fairly, at least in this sort of complexity of genome, with a lot of curation, but we can fairly accurately call these mutations. They're a major component of the mutation spectra, at least in Chlamydomonas. And transposons overall, when you include their higher rate relative to the other structural mutations and their role in mediating other structural mutations, were involved in around 4 out of 5 large mutations. And finally, basically, the transposons that were there essentially had a major impact on the spectra of structural mutations that we saw. If you had these kryptons, you had these translocations. If you didn't, in the other strain, we didn't see any translocations at all. So the transposons that were there were really important for the full spectra of changes that we saw. So now just to talk about some unpublished work, and this is all still using the same sort of experimental data, and it's very much a work in progress, but hopefully it's interesting to share with you. What we wanted to do is we had all of this Illumina data from sort of getting on for 8 years ago now, and we wanted to essentially ask, well, it would be really interesting to look at the structural mutations in these other ones, and of course it would be great to do long-read sequencing, but can we use these short reads that we already have to call some of these structural mutations? Now, the key thing that allowed us to sort of do this is Chlamydomonas. At the moment, there's an effort to make a pangenome. So we've started sequencing genomes for many of these different genotypes. And I think really here you want that sort of reference, ancestor-specific reference genome, because there's so much variation among those genotypes and a lot of structural variation. It would be very difficult to call mutations accurately. So we've been sequencing genomes. Unfortunately, I'm missing 2 of them, or we've got the genomes, but we haven't curated everything yet. But I'll be able to show you now 2 extra genotypes, and then also Chlamydomonas incerta, the other species. And I'm not going to go through all of this, but I'm sure anyone who's sort of called structural mutations or thought about structural mutations with short-read data is familiar with this sort of concept. Obviously, if you've sequenced the genome, it becomes much easier. You're just comparing one genome to the other. When you have short reads that are much shorter than the mutations that you're trying to call, you have to look for all sorts of, in some cases, obscure signatures that allow you to sort of infer a structural mutation has occurred, or a structural variant if you're looking at population data. So sort of split reads, for example, is one of the classic ways to do this, coverage differences, and so on. So we've been applying a number of different tools. So we have our sort of strain-specific reference genomes that we've assembled that we then map the Illumina data to, and then we use a number of different structural variant callers. Many of these are actually not focused on transposons. Many of these are looking for duplications, deletions. We are interested in the full spectrum. For today, I'll just talk about transposons, which is mainly using this MELT tool, which we give a transposable element library to. And actually, the transposons are so diverse that this required some iteration, because often the transposons we saw jumping in these other strains don't even exist in our reference set. So we had to sort of find them, curate them, add them, and run it again. It would be nice to extend this. There are lots of different tools that do this, and I think kind of methodologically, this is a fairly interesting data set because we have quite a good truth set. So for learning more about the sort of limitations of different methods, it would be nice to sort of extend this, and that's definitely something we're planning to do. But for now, I'll just be showing you mainly the results from MELT. Some of the other callers did contribute as well to these transposable element insertions, and they're all manually curated. So again, we get all the calls, we go through, we look at them. So we're fairly happy that these are genuine. So again, just the PacBio data, that difference that we saw between those two genotypes. So if we go and then we look at the Illumina data, how does it look? Well, perhaps that's not really that surprising. So it goes down by about half for one of the strains, and it's about 40% reduction for the other. And just kind of verbally sort of explain what's going on here instead of showing data, just given time constraints. But essentially, there's two things. One of those very complex mutations that I talked about, it's very difficult with Illumina if you've got a rearrangement and an insertion to sort of decipher what's going on. The other is that around 20% of the genome is highly repetitive, and Illumina reads just aren't going to map very well there. And not only is that the case, but if we look at the PacBio data again, this is just the different site classes in the genome, and the observed are the bars and the expected are the sort of whiskers and points. You actually see that transposons, and this is quite well known often, preferentially insert within intergenic sequences. So it's not just that there's 20% of the genome that's repetitive, it's that the transposons are actually going to those regions more often due to their own biases, due to their own sort of insertion biases, while deletions and duplications had a essentially random distribution relative to site class. So what about the other strains? Well, again, we see extensive genetic variation. So we've got really quite variable rates of transposition rates among these different genotypes. And of course, it would be nice to have more genotypes here, but one interesting thing is that this doesn't necessarily correlate with a single nucleotide mutation rate. So you can see for the CC2342, CC2344, it's actually the other way around. One has a higher transposition rate, one has a higher single nucleotide mutation rate. I wouldn't say that's really surprising given that the genes that underlie mutation rates for these different categories are probably quite different. And just the final slide, the transposition events, the spectra of different transposons, as we showed with the PacBio, this extends to the Illumina. When we look at our two other strains, completely different transposons that are active. So the size of the ball is just a number of events, and then different colors correlate to different types of transposons. And this is actually, we only saw one common transposon between all of these. And this is essentially expected based on what we know from Chlamydomonas. There's an incredibly diverse repertoire of transposons. Most of them are very young, which is what the x-axis is showing here. So we think that there's lots of different transposons in the genome that are generally silenced at any one point, but then in different strains, we can see different ones that are becoming active. So again, just to finish up and summarize here, simple TE insertions, so not the complex ones in non-repetitive regions, we have a pretty good handle on. So it would be interesting if we can do better, but maybe the complexity of the data is really the limit here. And we have extensive variation in transposition rates, as we do in single nucleotide mutation rates. These are not correlated, so there are presumably different things going on in terms of the genetic architecture underlying this. And essentially, for the different genotypes that we see, we have completely different transposons that are active. So this was all done with a postdoc at Edinburgh, Eugenio Lopez Cortejano, and I'd just like to thank previous supervisors who were involved in those very laborious experiments. And yeah, thank you very much.
Cool. Hi, I'm Rowan Green, and I'm going to be talking about how ecological and genetic factors can control microbial mutation rates. So this talk is kind of in two parts. The first half is more ecological, and the second half is more genetic. So when we think about evolution, and I mean, I'm sure I might have missed out your favorite part, but this is basically how I was taught it. We have random spontaneous mutations, introducing variation, and then selection imposed by the environment along with drift determines which of those genotypes will reproduce and survive into the next generation. But there are actually a wide variety of environmental factors that can change the spectrum and the rate of spontaneous mutations. And so we can term these plasticity. These aren't mutator or anti-mutator genes, they're just impacts of the environment on the type and rate of mutations. And for the first half of this talk, I'm going to focus specifically on the effect of density. So it's previously been shown that across microbial domains of life, as you increase density, you decrease your mutation rates. But what wasn't known was the mechanism behind this. And by better understanding the mechanism, I think we can have more of an idea about how this might be affecting the course of evolution, this change in mutation rates. And so what we started with was doing some ordinary differential equation modeling, and this allowed us to explore lots of different hypotheses in a much quicker way than measuring mutation rates in the lab, which is quite slow. And so the main conclusion we got from our modeling is that really the model needed to have density dependence in the production or the degradation of reactive oxygen species in order for us to then see density dependence in mutation rates. And this model, I won't go too much into it, but it simulates an E. coli batch culture where they take in glucose, they make nucleotides. Sometimes those get damaged, and so sometimes they have mutations. So reactive oxygen species are things like hydrogen peroxide, which can degrade into this hydroxyl ion, which is very reactive, and when it damages nucleotides, it can change their shape, causing them to mispair. For example, it makes guanine really happy to pair with adenine, which is not good. So then once we had these predictions, we had to go into the lab because the model gave us a nice idea, but we needed to prove it. And so these experiments I'm going to show are all in E. coli. And the first thing, which is just a nice correlation really, is that if you look in black, you can see the mutation rate dropping as density goes up. And if you look in red, you can also see that the external hydrogen peroxide in like normal lab media is also dropping with a similar type of slope. And so this indicates that it could be a change in peroxide driving this because the peroxide is changing. And so what this second graph shows is that on the y-axis, we've got the log-log correlation between mutation rate and density. So here is our wild type, and it's negative, which means higher densities have lower mutation rates. And what we found is that when we grew them in anaerobic environments, and E. coli is very useful in this, it's happy either way, then we actually reverse that pattern. So mutation rates are going up at higher densities, and that is the pattern that's seen at very high densities in aerobic environments because of stress-induced mutagenesis. So rather than going down and then up once stress hits, in an anaerobic environment, they're just always more cells, more mutation rate. And we could also show this in a genetic rather than environmental way. So this strain is missing all 4 of the E. coli enzymes which can degrade hydrogen peroxide. And so what we showed is that if there's no oxygen around, and therefore a lot less reactive oxygen species in the anaerobic environment, or if you can't degrade them, you have a very similar effect. So there needs to be the production of ROS, but also the enzymatic degradation of ROS in order to see that negative slope. But what was really convincing is that when we add wild-type cells to the strain which can't degrade peroxide, then the more wild-type helpers that there are around, the lower the mutation rate of that strain which cannot degrade peroxide itself. And so we'd shown how one strain can complement another strain. This was all in E. coli, and we're now trying to expand it to look at how different species of microbe can impact each other. But I'm not going to show that stuff today. It's very unfinished. So what I've shown here, to come back to this picture, is kind of how the genotype will also affect your plasticity, and different parts of the environment will affect how those are affecting things. So there's kind of higher order effects, right? You change the environment, and now the density in the environment is having a different effect. But this second story is going to show how the genotype can affect not only plasticity, but also the underlying spontaneous mutation rate. So we're looking at a knockout of NUDJ. This is a Nudix hydrolase. And the reason we got into Nudix hydrolases is if anybody works with E. coli or a lot of other things, you might have heard of MUT, which repairs damaged guanines, and it's in the same enzyme class as NUDJ. So we went and had a look in that enzyme class. NUDJ, that it has various roles. It doesn't seem to be very clear yet. It's not very studied. But one of the things it does is control the nucleotide pool. For example, it can take guanine diphosphate and turn it into guanine monophosphate. So this could be the way in which it's having an impact on mutation rates. So along with a previous PhD student and a really great undergraduate mentee, we showed that the NUDJ knockout has a much, much lower mutation rate than the wild-type E. coli. And because NUDJ is not so responsive to density, this difference also changes a lot with density. At low densities, it's having a massive effect. At high densities, it's much more subtle. And so the reason that we were really excited to find this anti-mutator is in terms of thinking about antibiotic resistance, where mutation rates can be very important, then when we grow a culture, a low-density culture of the wild type, about 88% of the time, in the absence of selection, we'll get some spontaneous resistance mutations. But in the NUDJ strain, that's only 31% of the time. And so this raised the question of, could a NUDJ inhibitor, making the wild type look more like that blue bar, work as an adjuvant alongside antibiotics to reduce mutation rates? But before we go down that line, we just wanted to understand more about the basics of what NUDJ is doing. Because as most of the talks earlier have touched on, a mutation rate isn't just a rate of one thing. It is comprised of multiple different mutational classes. We do have indels, but I'm just going to focus on single nucleotide base pair substitutions to keep things simple. So there are 6 different ways in which you can change which nucleotide you've got at a site. And so we sequenced lots and lots of our 2 strains, and we found that removing NUDJ not only reduces your mutation rate, but it also changes the types of mutation you're getting. And what was really interesting to us was a significant reduction in A to G mutations. So everything is being reduced, but A to G is being reduced a lot. I know that A to C here is proportionally going up, but obviously its overall rate is still down. And so this really neatly answered our question of why NUDJ is less responsive to density. So this similar graph here is from a previous paper by Dana Gifford at Manchester, where she showed that A to G mutations are particularly elevated at low density. And if NUDJ doesn't do many A to G mutations, even if they're elevated at low density, we're not going to see so much of a change in the overall mutation rate. And so hopefully, overall, I've shown you how this is a really nice system for understanding the multiple different factors that are altering mutation rates and how mutation rates are really very, very dynamic in a way we don't often think about. And so you've got effects of the genotype, you've got effects of the social environment in things such as density, and you've got effects of the chemical environment. For example, is there oxygen present? And not only do all of these alter your mutation rate and spectrum, but they also interact with each other. And so with that, I'd like to thank my supervisors and all of the Microbe Evolution Research Group in Manchester, which is an amazing environment to work in. The first story here got published 2 or 3 days ago in PLOS, so if anybody wants to look at that, the QR code does go to the real paper, not a preprint now. And yeah, if anybody has any questions, I'd love to hear them.
Can everyone hear me okay? Okay. Good morning, everyone. Thank you for being here. I'm Bryan Gitschlag, for those of you who don't know me, and I'm a postdoc at Cold Spring Harbor Laboratory. And the research program that I'm working on building is guided by an interest in the question of how do different, forces of evolution combine to give us the distribution of evolutionary outcomes that we see. And so, for example, you feel free to ask me later about my multi-level selection work. But for today's purposes, we could consider the question of how do mutation and selection combine, as we've already heard about quite a bit from some of the fantastic talks earlier. And more specifically, what role do mutations play in guiding the direction of adaptive outcomes? And so this is a focus of a project that I'm working on with my postdoc advisor, David McCandlish, and our collaborator, Arlen Stoltzfus. And so to kind of illustrate what I mean, I'd like to start by offering a few examples. And so, for example, if you were to mutate the alanine amino acid to valine at position 63 of the alpha hemoglobin, that has been reported to contribute to an elevated oxygen affinity and therefore adaptation to higher altitude, in particular in the bar-headed goose shown here. Likewise, if you mutate the alanine to a threonine at this position in the green algae Chlamydomonas, that's been reported to contribute to an elevated growth rate under conditions of prolonged light exposure. Or if you mutate the valine to isoleucine in this position of the HIV protease, then that contributes to resistance against various types of antiretroviral therapies, and in particular protease inhibitors. And the reason that I'm sharing these examples is because the one thing that they all have in common is that in each case, they're driven by a G to A mutation at the single nucleotide level. And it turns out that in most of the species that we've looked at, this tends to be the most common type of single nucleotide mutation. And as I'm sure you all know, because we've heard about it already, when one mutation occurs at a higher rate or more often than another type of mutation, we refer to that as mutation bias. And although these examples that I've shared so far are anecdotal, they do raise a couple of interesting questions. And so one question that we're interested in is, what is the relationship between the strength of a mutational bias and the bias in the distribution of adaptive outcomes? And a similar question that we might be interested in is, do differences in mutational biases between species drive differences in adaptive outcomes between those species? And so to begin addressing these questions, it's important to keep in mind that adaptive evolution is a process that combines a couple of processes, in particular mutation and selection. And so we can consider the question of what happens if there are multiple different adaptive paths. And in this scenario, the thickness of the arrows is meant to represent the relative mutation rates. And so what you see here is that the blue arrow actually leads to a higher fitness peak, but the green arrow leads to a more mutationally accessible fitness peak. So depending on the biases at play, mutations may conceivably drive a genome toward a fitness peak that is mutationally favored, even if it's selectively suboptimal. And this is actually consistent with previous work, which has reported that the most frequently observed evolutionary outcomes are often not those that have the greatest fitness benefit. And so we can ask the question, what sort of relationship should we expect to see between mutational biases and the bias in adaptive outcomes? And the answer is that it actually depends on some population conditions. For example, in particular, mutation supply. So when mutation supply is very low, then typically what happens is that the next adaptation that goes to fixation is going to depend on the next adaptive mutation that comes along in the population. And in this case, we would expect that mutation bias would have a very strong effect on the distribution of evolutionary outcomes. Alternatively, when mutation bias is very high, then we're in a situation where typically there are multiple different mutational variations in the population at any given time. And so in that case, we would expect that the fittest mutations would go to fixation, and in which case mutation bias may have little to no effect on the distribution of evolutionary outcomes. So in other words, the population genetic theory doesn't really predict a priori what we should expect to see in terms of the relationship between mutational biases and the distribution of evolutionary outcomes. And so for that, we need to turn to empirical data. And to that end, my collaborator, Arlen Stoltzfus, and I have been curating a large data set of individual adaptive mutations, and these come from experimental evolution studies as well as patient samples. And to help ensure that the mutations that we're considering are mutations that can be compared against measurements of mutation rate, these mutations in our data set have to meet a couple of important criteria. So for example, we're only considering for the purposes of this project, protein coding changes, and we're only considering protein coding changes for which a single underlying nucleotide change is known. And then furthermore, we're considering mutations that, for which there is empirical evidence of an adaptive effect. For example, studies that, functional studies that link a particular mutation to a particular adaptive phenotype of interest, such as drug resistance. And then furthermore, we're only considering species for which there's a large number of adaptive changes. And the data set that we have so far consists of more than 6,000 individual single nucleotide changes in proteins across 15 species. And in parallel, most importantly, we also obtained previously published estimates of the mutation spectra for these same species. And this comes primarily from mutation accumulation experiments and patterns of neutral variation depending on the species. And so what this allows us to do is compare empirical adaptive spectra of adaptive changes with empirical measurements of the mutation spectra for the same species. And of course, as we all know, there are different ways of classifying individual single nucleotide changes. So for example, we might be interested in classifying mutations in terms of transitions versus transversions. And so just to remind everyone, a transition is a mutation that preserves the purine or the pyrimidine nature of the nucleotide, whereas a transversion is a mutation that swaps between purines and pyrimidines. And so what we find when we look at the data is that transitions tend to contribute much more strongly toward adaptive outcomes, particularly in those species where transitions occur de novo at a much higher relative rate. But of course, there are actually 6 different ways of categorizing individual mutations, individual single nucleotide mutations. So we can ask the question, for each of these 6 types of single nucleotide mutations, what is its frequency among new mutations in a given species? And then, how does this, most importantly, how does this mutation spectrum compare to the spectrum of adaptive changes within the same species? Now, I should mention that a similar type of analysis was published previously by my collaborators a couple of years ago, where they looked at 3 species, E. coli, Saccharomyces, and Mycobacterium tuberculosis. And what they found was that within a given species, the types of mutations that arise most often tend to be the most commonly observed type of adaptive changes as well. However, what we've been able to do with this expanded data set is we've expanded the state of the field to include 15 species, for which each of which we've paired the mutation spectrum with the spectrum of adaptive outcomes. And so this includes the 3 species that I mentioned previously, plus 12 additional ones. And again, the general trend is that in most species, what we see is that the mutations which occur at a higher rate tend to contribute more toward adaptation. But what we want to do actually is take this relationship between the de novo frequencies and the adaptive frequencies of given mutations and compare this relationship across species. And that is, in order to answer the original question of, do differences in mutational biases lead to differences in the distribution of adaptive outcomes between species. And so one way one sort of rudimentary analysis, I should say, to help get at that question has been to combine all 6 mutation types. I apologize, that's not really showing all the mutations. In any case, all 15 species, and we've combined all 6 mutation types across all species. Can anyone see that? Not really. There's a positive correlation, I promise. But what this plot does is it collapses all 15 species of the within species all 15 of the within species comparisons, that we just saw a moment ago into one plot. So I'm just going to skip ahead because you can't see the individual mutations. But since that was a plot that compares both within species and between species, in order to really isolate on the comparison between species, what we've done is take the same data, the same scatter plot, which now you see it here is we've taken the same scatter plot and we've isolated the analysis to a single type of mutation. And so the colored data points now are the same mutation, but all 15 different species. And when we perform this comparison across all mutation types, we find that the regression is highly significant. Now again, there's some gray lines, there's some positively correlated gray lines that you should be seeing that you're not. Those are bootstrap replicates. The significance test here is coming from comparing the actual data against simulated data under the null model in which mutation bias has no effect. And so what we see is that, again, for a given type of mutation, if that mutation occurs more frequently in one species than in another, it also tends to contribute more toward adaptive outcomes in that same species. And so to bring it all back home, what we find is that new mutations that occur more often or at a higher rate, they tend to be stronger contributors towards adaptive outcomes. And we find that to be the case both across mutations within a species, as well as when we're comparing across species with respect to a given type of mutation. Moving forward, I'm continuing to work on other ways of studying how mutation and selection combine. And in particular, my collaborators, David McCandlish and the other members of the McCandlish Lab and I are developing methods for modeling the distribution of evolutionary outcomes by modeling the effect of mutation bias in high-dimensional fitness landscapes that contain millions of individual genotypes. And I'd be very happy to talk more one-on-one about the nature of this project if you're interested. But in the meantime, I'll go ahead and wrap up by thanking the organizers of this symposium, of course. I'd like to thank my collaborators and colleagues in the McCandlish Lab and Arlen Stoltzfus, our funding sources, and of course, last but not least, I thank you all for your attention, and I'd be very happy to take any questions at this point.
Okay, thank you all for being here. Today I'm going to be talking about the mutation rate in possums. And as I'm sure most of you already know, the mutation rate itself is a trait, and just like any other trait, it evolves over time. And therefore, we can observe a lot of variation, not only across different populations, but also even within across different species, but also even within populations. And in fact, the mutation rate varies by more than an order of magnitude about eukaryotes and around twofold among primates. Today I'm going to be focusing on those mutations that happen in the germline, which are also known as de novo mutations. And what are these mutations? Well, these are variants that we can see in the offspring despite not being observed in either of the parents. And this is basically how we detect them. After sequencing the genomes of all the individuals, we focus on those sites where we observe a variant that wasn't present in any of the roots of the parents. And this has helped us to see how many mutations each of us or any other individual can inherit or can be born with. In humans, for example, we know that every person is born with about 70 mutations, which accounts for a mutation rate of 1.2 times 10 to the minus 8 per base per generation. But as you can see, there is a wide variation in the number of mutations every individual is born with. And there are different factors that can contribute to this difference, being parental age one of the most common or the most talked. And why is this important? Because as you can see, as parents, in this case fathers in particular, as they age, the number of mutations also increases drastically. At this time, or what I'm presenting here is a study from 2012, where it wasn't possible to see whether mutations were coming from mom or from dad. And therefore, they cannot include like a parental age in general. And they decided to only focus on the fathers. But now, things have changed, and it is possible for us to see whether mutations are coming from mom, if they are coming from dad, and if we observe the same pattern. How do we do this? Well, after identifying our mutations in families or in pedigrees, we identify the haplotypes that are carrying such mutation, and then we trace that haplotype, and by doing that, we know which parent the mutation is coming from. But this is only possible when we have multi-generational families. That means that we have to have at least 2 generations. The other option for when we don't have many generations, or even if we do, we can use, it's by read-based tracing of these de novo mutations. And what I'm showing here is like in the last set, you can see the mutation, which is represented by a star, linked to a SNP or an informative site. So that SNP is going to help us to identify the origin of this mutation. If we know that this SNP is coming from a particular parent, then it's also possible to know where the mutation is coming from. And this was very helpful because, as you can see here, this allows us to not only to see that the paternal age effect is very strong, but now we also see a maternal age effect. It's not as strong as the one coming from the fathers, but it's still there. But most importantly, we observe a very strong male mutation bias. That means that most of the mutations that individuals are being born with are of paternal origin. In fact, about 75% of all the mutations are of paternal origin. And this kind of makes sense if we focus on what we have always been told, if we focus on the replication origin of mutations, right, or the textbook explanation, where we basically know that in females, the germline stops dividing right after birth. And this is also something that happens in males. But the difference is that after reaching puberty, males continue to divide their germline. And therefore, the more cell division, the more replication, DNA replication cycles, which is going to end up in multiple errors that later on are going to be mutations, and these mutations are going to start accumulating. Well, recently, we have encountered different findings that are very inconsistent with the textbook explanation for the male bias mutation. Here, in this paper that we published last year, we talk about 6 of these inconsistencies, where we basically summarize everything to the fact that this bias that we observe is due to a lower protection or repair machinery, inefficient repair machinery in the germline of males. So is there any male bias? Oh, and then this led us to our next, today I'm just gonna be talking about this question. Is there a male mutation bias even at very early parental ages? And why am I saying this? Because as you can see again in the diagram, like if we focus on parents that are very young, we can estimate a similar number of cell divisions, right? Or similar number of cycles of cell divisions. It would result in a very similar number of mutations. But that's not the case. When we look again at the human data, as you can see, the male bias is already present at very early ages. And in fact, this male bias, or this ratio of male versus female mutations, it's always the same. It keeps constant across different reproductive ages. So, and you might think, okay, maybe this is happening because of the long time that humans have to wait until they become mature, sexually mature. So, yeah, we decided to test this in organisms that start reproducing very early in life, and we chose the possum. This is not the possum that we sadly saw on the road. This is the little possum from South America, which is a very interesting system for us because they diverged from the eutherian mammals about 160 million years ago, which is enough time to accumulate differences. They also have unique developmental traits, including a shorter generation gestation periods and very early birth. And they possess unique genome characteristics, including the lowest recombination rate reported so far and a very low CG content. Most importantly, they reproduce very early in life at about the age of 6 months to 1 year. This is the data set that we were looking at. Our data set is conformed by 3 extended pedigrees with 22 trios, which accounts for 34 individuals. And this is basically what we did. We sequenced the genomes of every single individual, and then we aligned our samples. We then called for variants, which led us to the identification of mutations, and finally we phased these mutations. Or with phasing, I mean like we identify the origin. So we found a total of 246 mutations across the 22 trios, 4 of which were multinucleotide. The average mutation rate for this species was 0.256 times 10 to the minus 8. And this is actually the lowest mutation rate reported in mammals so far. And you might ask, like, how low is it? Well, this is, again, the human mutation rate across different ages. And the possum data looks, I don't know if you can see it, the green dot. It's very, very low. But we use a reproductive longevity model that is very useful for us to estimate other mutation rates in other primates. And if we use that model, then it doesn't look that different. So basically, possums are little humans. And then we also classify every single one of our mutations, and this is how the spectrum looks like. Overall, it doesn't look very different to the spectrum of other mammals. Most of the mutations happen at where C to T transitions, and the mutations happening at CPG sites were overall like 22% higher, 22 times higher than the overall mutation rate. Again, not many differences between the one in possums versus the humans, but we can see a very clear difference, and that's how many of these C to T transitions happen at CPG sites. And it's actually half of the one reported in humans. That was kind of weird for us, but I told you before that one of the characteristics of possums is that their genome is kind of weird, right? And in fact, they only harbor like half of the CPG sites that humans do. So after kind of considering both things, we corrected for those differences, and we really didn't find so much difference. Then we wanted to assign every of these or most of these mutations to a parent, and I told you how we do this, right, with these 2 different methods. So we went ahead and took advantage of these 2 different methods, and we created this program, Possum Hab, which stands for Parent of Origin Sex-Specific Mutation Haplotyper, which is very helpful for the identification of the origin of mutations. This is soon to be released with this whole project. And this is what we found. Of all our mutations, we were able to identify over 50% of them, or the origin of over 50% of them, 72% being of paternal origin. So again, very consistent with what we have seen before in other mammals. And this is basically the same, but represented in a different way, where you can see how the male bias is always very strong, but what makes our results a little bit different from the ones reported in other mammals is that we didn't observe an age effect, as you can see here. And yeah, we were kind of disappointed. But then I was thinking like, oh, maybe this is happening because we don't have enough, a wide range of ages, right? There is only 1 year of difference between the youngest one and the oldest father. So we decided to kind of simulate the possum information using human data. And this is, again, the human data. And I was creating different intervals with 1 year of difference to see if we observe an age effect when there is only 1 year of difference. This is, oh, I guess you cannot see very clearly the lines, but like I ended up with 44 intervals. And then I tried to see if there is a parental age effect. And it was surprising that only 7 out of the 44 different comparisons that I had showed parental age effect, also in a very big, very large sample. So this is not significant. And this basically tells us that the fact that we are not observing the same in possums doesn't mean that there is not an age effect. We simply cannot test that because of their lifespan and how early they reproduce, how early they die, how early they stop reproducing. Unfortunately, we're not gonna be able to test this, or maybe we could if we only sequence like probably thousands of them. So what are some possible explanations to the male bias? Well, it could be, as I mentioned before, that there is a very inefficient DNA repair machinery. There could be also some sex-dependent differences that are responsible for this, including like sex or hormone-dependent mechanisms, the gene expression in the germline of the two sexes, and of course some metabolite production and accumulation between the two sexes. So these are just the different points that I went through. We found the lowest mutation rate so far reported for mammals. Despite this, we observe a very, very strong male bias. And also, we think that what we are observing might be another indicator of why the males can be considered as faulty, because there is definitely something going on with them. And with that, I would like to thank the university, Indiana University, my amazing PI, Matthew Han, and Richard Wang, who's a postdoc in the lab and who was very helpful, and of course, every single other member of the lab. And with that, I will be happy to take any questions.
Hello. So my name is Daniel Sudd. I am a postdoc in the Chen Lab at the University of Rochester, and today I'll be talking to you a bit about how we're working on it is auto-transferring. Working on how we're investigating mutation rates in low coverage pedigree data. So as you've heard from multiple talks at this point, unless you just got here, mutation rates vary in a wide variety of complex ways due to a lot of factors, not even just across taxa, but even within individual species as a result of factors that include but are not limited to mutator alleles, parental age, although apparently there may be some pushback on that. I need to read some new literature, and even environmental factors. But long story short, there happen to be, what is happening? Okay, long story short, we generally need to improve our understanding of we need more data points, so to speak, to complete our understanding of how these rates vary across taxa. This is for some reason very rapidly auto-transferring from slide to slide. Okay, I'm not sure why that was happening. Key combination that goes straight into presenter mode. It's not, the screen's not duplicated, I can't. Let's see. Okay. All righty, I guess I'll make do with that. In any case, so as many of you may be familiar with traditional de novo calling methods, wherein you're dealing with a series of closely related individuals, frequently parents and children, that have high coverage across the region of the genomes of interest, and you are looking for read consensus with relatively high read depth to see if you can find candidate mutations in order to actually make these calls. This is very effective. However, it can be expensive when you're dealing with large sample sizes. So while there aren't a huge number of truly massive available pedigree data sets yet, there are some, and people are still working on gathering them in the future. So our own lab has access to and is currently working on a truly massive 4,000 sample pedigree for the Florida scrub jay that I hope to be investigating. But even beyond our own personal investigations, I'm hoping to create a tool that enables further investigations of types of pedigree data that previously were not really practical for these types of mutation analyses due to the prohibitive cost of trying to sequence all of that to high coverage. And also, being able to extend into mutation calls across greater numbers of individuals potentially allows avoiding some specific biases for the smaller number of samples that might normally be viable for these kinds of analyses. Our weapon of choice is IBD segments, identical by descent. And what that implies is that you have two sequences, sometimes in two haplotypes, sometimes in two individuals, that are nearly identical as a result of sharing a common ancestor. And that nearly is very important for mutation analyses. Now, we are not the first method to be attempting to employ IBD for mutation calling, but we are leveraging it in some slightly novel ways. And in this particular instance, we're taking advantage of pedigree structures. So if you just have pedigree data, what does that tell you? It gives you a set of relationships between samples in question. So you get a general guess, since you know if you're dealing with a diploid organism or a polyploid organism, you know that recombination will allow a certain chance of regions of the genome to be inherited at each successive generation. That's good, but it's nowhere near enough to be able to actually localize mutation information of the form that we're discussing. If you continue to add context, though, if you perform IBD calling, you now have an ability to tell what regions of the genome actually are present and shared between these individual organisms. And having pedigrees helps improve the accuracy of these IBD calls in addition. So that's also, that comes together quite nicely. So that additional piece of information comes together even more when you start trying to perform sequencing. So if you aren't trying to perform high coverage sequencing at all the samples in these pedigrees, you'll end up with you may end up with low coverage or variance depending on how you've chosen to go about your study. And in this particular instance, we are working with mostly low coverage with some interspersed high coverage samples at informed locations. But for here, our focal individual, which has a de novo mutation candidate we're trying to investigate, that individual is not high coverage, nor are its parents. But you might notice that there are still a lot of reads available from the surrounding pedigree. We can, in fact, split our pedigree across the parent-child, the parent-child barrier between our sample where we're trying to call a de novo mutation, and take advantage of that almost in regards to how IBD segments are almost truly identical, because those differences are where mutation events occur. So if the event that you have an IBD segment with the region where the mutation is present, you can make the assumption that any IBD segment that is a descendant of that region will show evidence of a true mutation, and any IBD segment that is ancestral to or IBD through an ancestor to that region should not show any evidence of the mutation in question. So with that assumption in mind, you can effectively pile the reads from the region that you are making the IBD assertion for, and build a proxy high coverage parent and the proxy high coverage child to perform an analysis that is very much like the traditional high coverage methods that you had that are very popular and very expensive for large sample sizes. So there are obviously caveats here. As you've learned a little bit from the last talk, phasing, which is a key step for IBD analysis, and the IBD analysis itself, they're not free. There are a wide variety of ways that they can go wrong, and you can end up with miscalls. So if you are trying to create an algorithm that is robust to a wide variety of pedigree structures of variable coverage, ideally it needs to have the ability to handle when you can't get some of these data forms in certain locations within the pedigree. So we are attempting to demonstrate the algorithm we've produced is capable of these under many backgrounds by performing simulations to stress test it in various ways. In this case, our input comes from a series of background variants that will potentially serve as false positive counts, or false positive candidates, that we create via standard PopSim. And although for the results I will be presenting today, we are exclusively using rare variants rather than the full set that would be coming out of that. As input, we take a pedigree structure. This is a stand-in for whatever pedigree of interest the study would potentially be employing. And the coverage strategy, either the coverage strategy that was already decided and trying to confirm what can be done with what's already there, or as a hypothetical coverage strategy if someone hasn't done the study yet and is trying to make some decisions on regards to what can be used to actually still get mutation calls of an adequate power and an adequate precision out of that. So we put that through MS Prime. We use that to simulate recombination rates. We use that to simulate mutation rates. And we effectively fill out the pedigree with proxy genomes that function for most intents and purposes like the real genome would. We then introduce our error metrics to make it so that the problem is less free than it could otherwise be, try and make it a little bit more of a realistic task. So that includes errors with IBD inference, errors with phasing, errors with sequencing. And with the now sufficiently muddied data set, we perform our evaluation. So for this particular presentation, I'm going to be talking about two types of results that we were dealing with. The first is variable coverage strategies. I'm showcasing 3 examples here to get the intuition across. It's not a full set of the ones that I'll be showcasing in the results, but the idea is, as you go from left to right here, and as you go from left to right on the x-axis on the next slide, we will be going from basically max high coverage, where all the samples are 20x, to a midpoint where we are removing the internal generations from the high coverage pool. So at that point, it approximates 70% coverage down to successive decreasing quantities of the final generation, where we stop at approximately 17% of the samples being at 20x coverage, with the remainder, which are here filled in as the white circles, are down to 2x for the particular tests that we are running. So our evaluations here are power to be able to actually call the mutations in question, and precision, wherein we are avoiding rare variants coming from our background set being miscalled as mutations. So the power does indeed drop off as we go from left to right. And this is somewhat inevitable because, and I looked into the data and checked, there are mutations as you drop your coverage that just effectively disappear from the reads, and you can't call a mutation if there literally isn't any evidence that it still exists in the data. So eventually, as you keep dropping coverage, you are going to lose some of the mutations. That's inevitable. But apart from that, the statistics that we use to avoid classifying mutations as non-mutation variants are fairly effective, even all the way down to our minimum coverage example for this test. And with regards to miscalling the rare variants, so there are actually some miscalls in tests other than the lowest coverage example, but the fractions aren't visible in this particular example. With regards to, it's the patterns of reads that fall, even with the errors involved, just do not show, just still provide enough evidence to make these classifications. But I will admit that these tests are being run with somewhat lenient thresholds that are favoring power. There are alternatives tests that can be run that will not showcase it quite as well, but it still gives a good illustrative point for how the performance of this varies. For this particular test, we are now varying amount of awareness of IBD. So as we go from left to right, we are basically dropping the IBD information from successively larger numbers of the low coverage samples from approximately the midpoint test of the coverage test examples. And while our power remains very robust across all of these, the choices we have made lead to there being a bias towards calling things as mutations. And as we start dropping more and more amounts of IBD for more and more samples, it does become an issue where we start having miscalls. So I still need to further some investigations on different thresholds to confirm some of the trade-offs. But for the moment, it does appear that IBD segments, as we're expected, are rather important for these analyses. So many of these tests are ongoing. I have a lot more tests to run. I have more background pedigrees to use, but the punchlines for what I can show today already are that yes, even with very low coverage for a fair number of pedigree structures, it is very possible to be getting relatively reasonable amounts of mutation calls. And because of the way the dropouts in coverage are done here, you actually can predict how much of the region of the pedigree is available, which makes it possible to account for the fraction of mutations you can expect to call. So you can go from that to a successful mutation rate, even if you aren't necessarily able to call the mutations in all regions of all samples in question. So that is salvageable and remains useful for rates and spectra analyses. But it is still sensitive to dropouts in IBD, which makes the choice of different IBD algorithms very important. I am going to continue investigating individual IBD tools to see what their error rates are across different pedigree backgrounds. In addition to that, I need to finish my ROC curves for the thresholds to confirm the trade-offs, but they will be different on different pedigree backgrounds. So it's not just a one-and-done plot in that case. It's a lot of work left to do. We are hoping to do some human investigations where we're going to take values from the literature for mutation rates and see if our algorithm can really recapitulate those. So we're working on that. And we have that ScrubJ data set that I was mentioning earlier. We're going to actually use it to fill in one of those data points that I was saying we need for the ScrubJ. We're going to try and get its rates. We're going to try and get its spectra, ideally unbiased by being localized to a small number of individuals, since we're pulling from a relatively large pedigree. And we're hoping if we can get enough power to even do some mutator allele association studies as well. Then I would just like to thank the other members of my lab, the funding source, the NIH, and all the people who go out in the field and actually gather data, because otherwise there's no point to my research. You are appreciated. Thank you.